and welcome to the inaugural meeting of the Imaging Core Leaders. We're delighted to have everybody here. My name's Sterling Johnson. I'm the Associate Director at the Wisconsin ADRC, and I'm the Biomarker Core Leader. And I'm joined by my co-chair, Beth Mormino, who is the co-core leader of the Imaging Core at the Stanford ADRC. And we're delighted to roll out the Imaging Core infrastructure to you today. There's been a lot of changes happening over the last few days and few weeks, and we're excited to roll that out, talk to you about it, and get your input on it. Let's go to the next slide. So the goals for today would be to share the NIA vision and the new resources that they have provided and that we're rolling out now for the imaging cores and the biomarker cores that have imaging. We'll describe the new Imaging Core Steering Committee that was mentioned earlier by Nina. And then Dr. Jagast and Jack will present the new SCAN infrastructure. Dr. Mormino will present the survey results. We'll talk about the proposed imaging protocols that have been discussed and thoughtfully vetted for probably about 3 years now. And I think we've come up with a good solution that we need your input on. And for sure, we need input on all of this to make sure that we're guiding this infrastructure in a way that'll be useful to all the centers. Let's go to the next slide. So this is just a slide of the members of our steering committee. These were elected by the Imaging Core leaders, and we all have temporary terms, and it's going to be rotating. And this is our committee together with the NIA program officials. Let's go to the next slide. So what is this committee going to do? Our main job is to represent the needs of the ADRC imaging cores and components to NIA and to SCAN, and to recommend based on the needs that we identify and the scientific direction that we identify, particular recommendations for SCAN. Will assist the NIA in achieving any applicable ADC panel recommendations. We're hoping that we can facilitate more synergy and research across the centers with some harmonized imaging, while protecting and encouraging your local innovations that you're doing with your imaging. And finally, we plan this annual meeting at the Spring ADRC Imaging Directors Meeting. Let's go on to the next slide. All of this, these new changes, the infrastructure that you're going to hear about today, it's going to take some coordination and communication between the centers and the resource administrators, and we need your input on all this to make sure that we're doing this in a way that meets your needs. So I'm excited to pass the baton to Cerise to talk to you more about this. But before I do, I just want to thank the NIA for making all of this happen. There's been some incredibly positive changes that have really taken a lot of effort on their part. And finally, just to plug on the last slide that I have, to keep in touch during all of these changes as we roll things out with the new Slack channel. So before we go to Cerise to really talk about that, any questions at this point? Otherwise, we can turn it over to Cerise.
Thanks, Sterling. So we're just going to give a little background, a little bit about everything. You can go forward, Elizabeth. The first thing that all imaging core leaders should know about is that there was the administrative supplement call and the overall goals and the things that you must have in your application. And Bill and Cliff will tell you the instructions you need to do thumbs up of support. The awards are limited to $250 direct cost for a single year, and it may not extend beyond the end of the project period. And it's due on Friday, May 22nd. Next slide. I just wanted to go back to London, July 14th, 2017. This is when we did some information gathering about imaging. We started in April of 2017. We met in London at the AIC meeting, a big group of us, and everybody I think was represented, and we went over the costs and the benefits of standardization. Then we had an imaging meeting at the spring meeting in 2018 where we had a standardization proposal and presentations that are on the NET website that you can go and look up. And in May, we announced the U24 scan with RFA. In October, we established the Imaging Core Steering Committee that Sterling discussed and introduced to you. And today we'll introduce to you the scan PIs and get started on this work of scan. Elizabeth? So overall, in the neuroimaging core structure so far, there are 16 centers that have neuroimaging cores. There are 7 centers with neuroimaging within biomarker cores, and there are 8 centers with neuroimaging using other structures. Next slide. And in making a plan for the next 5 years, in tiling up what you talked about, there's a possibility over 3,000 scans across the whole center program that could be standardized and harmonized across the program. And that could be a data set for answering some important questions. You can go forward. So overall, I think that NIA, we have these overall imaging goals. We want to disseminate and utilize the clinically valuable data paired with the neuroimaging because we believe that it can advance the field. And we want broader dissemination and standardization of neuroimaging and the other biomarker tools that you guys do to diagnose AD and ADRDs. SCAN is funded to manage the sharing of standardized imaging data of the ADRC participants, the standardized collections of the MRI and the PET neuroimaging scans, and work with you to make a plan for incorporating the development of new MRI and new PET neuroimaging methods as they come along. They will work to QC and transfer and harmonize those standardized scans to a central repository, and they'll tell you more about themselves soon. NAC is responsible for managing the neuroimaging data in a repository and to share all of the data that you deposit. Next slide. This is our relationship model. Again, we're getting input from the centers through the Imaging Steering Committee, and NIA is managing NAC, SCAN, and the Imaging Steering Committee. So we're all in this relationship together, and we always want your input. Next slide. Just remember that the steering committees are always on the NAC website. There's always a call by Maggie to nominate yourself or nominate a fellow individual for elections. So these committee members have been elected, so remember that. And the next slide is to move your conversation to Slack, and that would be great. And I'm done, and we can get started on the amazing afternoon we have planned for you.
Thanks, Cerise. Are there any questions for Dr. Elliott before we move on?
And don't forget to input your questions in the Q&A box, and then Bill and Cliff are open to you guys raising your hand, and I can unmute you if you have really questions about that too. So,
Cerise, I have a question.
Yes.
For the supplements, do you know when they will be reviewed and what's the earliest funding date?
They'll be reviewed internally, and we have a process, and they'll be funded by the end of the year, the fiscal year. Are you there? think I'm losing.
Yeah, we heard that. Thank you. Okay, well, let's turn the time over to Bill Jagast, who will talk to us about the SCAN initiative. Go ahead, Bill.
Okay, great. So we're going to have 3 talks today. Actually, I'm going to just give you an overview of SCAN, and then actually 4 talks. And Beth and Sterling are going to explain the survey that they did, and then Cliff and I will come back in with more specifics about the PET protocols and the MR protocols. So we're going to get into the details a little later. This first presentation is just about the big picture of what SCAN is. So I can't take credit for the name. That was the title of the FOA. We just absorbed it and decided to run with it. I think it's a pretty good name. This is the overall goals of the program. As you've heard, it's to standardize imaging, basically. That's acquisition, quality control, processing, image analysis across all the centers. We want to raise the level of expertise to get everyone who wants to do imaging able to do it. The goal is to facilitate sharing of data and collaborative research. And we're talking here now about prospectively collected data, not data that might have been collected in the past. And it's MR, amyloid, tau, and FDG PET. So the next slide. So I'm sorry to get into the weeds right away, but I thought it would be good to go over the specific aims. And again, these largely reflect the FOA, and I'm going to talk a little more about how we're going to implement these, but I thought it would be good to start with this. So we're going to be interacting with NAC, with the NIA, and with the Alzheimer's centers, and one of our interactions is going to be working with NAC about uploading and de-identifying images to the image repository. We're going to develop and promote standards for how MR and PET data should be collected. Number 3 is to apply quality control on all the images. Next, develop and apply methods for harmonization, that is pre-processing of MR and PET images so that we can merge data across centers. And also we want to be able to merge data with data collected through ADNI. We want to make sure that all the data are curated, labeled, the status of their QC and pre-processing is available, and they'll be available for investigators to download the images themselves. We're also going to do data analysis for both MR and PET. We actually have a pretty ambitious agenda for that. So for MR, we're talking about brain volumes, cortical thickness, as well as for some of the images, vascular lesions, white matter hyperintensities, and integrity, cerebral microbleeds, perfusion, and then glucose metabolism, amyloid deposition, and tau deposition. So pretty ambitious. We'll get into that in more detail. Next slide. So I'll try to explain how we think about innovation, but we will certainly try to incorporate innovative developments as they develop and as they are sort of ready for best practices. And I'm definitely open to input on that. We'll be involved working with NAC to make sure all the data is linkable to other data on research participants. And then the next 2 are to establish mechanisms to make sure that all ADCs can participate in this regardless of their current expertise in imaging. So some sort of online help function and a website that will hopefully be helpful and certainly well documented. And then finally, I'll talk in a minute about how we envision the leadership of this group and its interactions with other stakeholders in this whole operation. So next slide. So Cliff and I are co-PIs of this grant, and probably, I mean, I think we'll both be doing a lot of shared work, but you can understand that I'll probably be more involved in the PET aspects and Cliff in the MR aspects. I'm working with Susan Landau and Suzanne Baker in Berkeley. Bob Kepi is doing the QC out of Michigan. And at Mayo, it's Cliff and Kajal, and at UC Davis, it's Charlie DeCarli and Pauline Maillard. So this is the essence of our team. Next slide. So you've seen this a couple of times, but I want to emphasize a couple of things about SCAN in the upper right-hand corner. We are not going to be giving the ADRC's marching orders with the very limited exception of our standards for image acquisition and and how we're going to do quality control and so forth. We look to the ADRCs as telling us what they want. And so for one example, in your upcoming response to the administrative supplements, we are not interested in telling you what kind of participants you should scan, or how long you should, how often you should have them come back, or anything about your scientific agenda. We hope in the future that the Imaging Steering Committee will bring the ADRCs together with common scientific questions and themes, but that's not our job in SCAN. Our job is to interact between the ADRCs and NAC and the NIA to make sure that the best quality data gets collected and standardized and managed in a way that it's available to everybody. So that's where we are on this. So the next slide. So this is kind of the overall structure. From the bottom up, the ADRCs will be collecting data. They're going to upload the data to NAC. At this point, there's going to be an interface between NAC and LONI that has not been precisely worked out. So I don't know exactly what that interface is going to look like. But hopefully, for those of you who have been uploading data, it's not going to be something that is going to be terribly different or certainly difficult. And so there's going to be this upload directly to NAC. Again, we're not involved in that. And then once the images are at NAC, they'll go down a separate streams for PET and MR. They'll go to Michigan for QC and harmonization, and then to Berkeley for analysis. And the MR images will go to Mayo for QC and harmonization, and then to Mayo and UC Davis for analysis. And on the top is the executive committee you're seeing, which is all of the key players that I already mentioned, as well as representatives from NAC, from the Alzheimer's Centers program. That is the steering committee. So that's that right now is Beth and Sterling, and a representation from the institute. Here you see, also on the right, an outline of who's doing what. So the next slide talks a little more about a little more specifically about the data flow. So again, on the left, users are uploading the data. Also, they're able to download the data. Oh, I didn't mention on the previous slide, data will be returned to the ADRC. Oh, thanks. That little pink arrow at the bottom, the results will return to the centers. And so how you want to use them is up to you. Back to the next slide. So yeah, so that arrow from the users to NAC is a bi-directional arrow because the centers will be uploading data and hopefully downloading data as well. And then there's the MR and the PET streams. You can see that at Mayo and at Michigan, there are early events in working with the headers. There's going to be a data form for all scans, which the centers will have to fill out and upload with the scans. And then the processing and pre-processing begins with the QC at Mayo, the QC at Michigan, and then the data analysis at Mayo, at Davis, and Berkeley. And then the results going back to the NAC database, again, linked to all of the other data on all participants in the ADRC programs. Next slide. So we have thought a great deal about this versus innovation. And we've been involved in thinking about this, as Cerise pointed out, since the very beginning. And there's been a lot of concern from the ADRCs about being told how to collect images when they are collecting images in ways that they feel are very innovative. And we understand that. So we really don't want to stifle a single center investigator-driven novel imaging research. But we do think that if we want to characterize research participants in this ATN framework, there are widely acceptable methods that are best practices and that we can all agree on, and that we don't think are going to stifle innovation if we propose them. And so in the immediate future, we're really going to focus on data acquisition and analysis that's related to this ATN phenotyping. We think that most centers, from everything we can tell from the multiple surveys we've done, we think that this will not be a problem for virtually all the centers. And then as novel approaches become available, they'll be incorporated based on the interests and needs of the Alzheimer's centers. Again, this is not something we are going to promote from on high. We're going to see what people are doing and what they want to do. And the goal, again, is to facilitate collaboration. So the next slide. So we're going to talk about the protocols in later sessions. Just as a sort of a preview, the MR protocols are going to have a basic required acquisition that's about 10 minutes long, and then optional additions. And for PET, we're going to use a fairly, I think, accepted standardized methods of collecting amyloid, tau, and FDG data in the frames that most people are already using. Next slide. So for MRI data analysis, again, you're going to hear about this in more detail, but at UC Davis, they're going to be doing the basic analyses of the basic data, which are cortical thickness, regional volumes, white matter hyperintensities. And then the optional sequences are going to generate data that's more related to what you see here under the Mayo label, which is cerebrovascular lesions, DTI, CBF maps, and resting state maps. But these are again, we don't know how many centers are going to opt for collecting these data. Next slide. For PET, again, sort of telegraphing what I'm going to talk about later, we're going to be doing very much, again, this is all very much based on what is happening in ADNI. We want the data to be mergeable with all the ADNI data for MRI and for PET. So this is really quite similar. There's some minor differences we can get into later. But we're going to be using FreeSurfer-based regions to characterize tracer uptake for amyloid and tau PET. For amyloid, we're going to provide data in centiloids, and we're going to provide positive, negative status based on the thresholds that we've worked out in ADNI. And then for FDG PET, we don't know how many people are going to want to collect FDG PET, so I'm not sure what the status of that is going to be in SCAN, but if enough centers want to do it, we'll also do similar analyses with FDG data. Next slide. So all the data are going to be uploaded. This is one of the things we'll talk about. I don't know how much, whether there are any regulatory issues at the level of the ADRCs for this, but all of the data are going to be available at NAC and linked to subject IDs, and also the quantitative data will be at NAC as well as the images. Next slide. So as I mentioned, we really want to enable imaging across all the centers, and so we're going to be working out how to establish protocols and provide facts and help, some sort of help desk. I had to stress not in real time. I think you probably know that, but we'll do our best to help the ADRCs that want to collect images but haven't had as much experience. Next slide. So this is what we see as the priorities. I think we're pretty close to the first in terms of finalizing the protocols. We will start working on establishing data collection. There's still a lot of things we haven't figured out. We just got funded last week, I think it was. And we're going to be working on the data flow processes in terms of how the data is going to go from the Alzheimer's centers to NAC and how the data is going to go from NAC to the analysis groups. But I think we have the time to do that. We'll then start with data uploading in a limited number of the test sites and then develop help functions. Okay, in the next slide. So this is something we can either talk about now or at the end. I think we built in a fair amount of time for discussion today, so we might want to keep this the discussion of this issue until the end. But let me tell you what we're thinking about how we would interface between the ADRCs and NIA for these administrative supplements. So we really don't want, so this is partly out of my desire to avoid work. I don't want to, neither Cliff or I want to write 30 letters of support. So we thought it would be much more straightforward if we circulated a document that outlined the substantive requirements of our protocols. And that would be how you're going to analyze acquire the data. One of the requirements is that you're going to have to use a single scanner for all images, whether that's an MR scanner or a PET scanner. You're going to have to pick that scanner and stick with it for all of SCAN or all of your ADRC. And I know that could be an issue we can discuss. You have to agree to the requirements for uploading images, which aren't really going to be that cumbersome, and making the data available. So that's really the core of what we want from the centers, and what obviously we have to have from the centers really in order to make this program work. And basically we'll circulate this document, and what we were thinking is having some sort of check boxes where you indicate that you will agree to these things and which precise protocols you're going to choose, and then submit that with your application. And that would be a indication that we would give you approval, as we give you our letter of support. So we don't want to know, like I said, it's between you and NIA what types of participants you want to scan, and what modalities you want to scan them in, and so forth. We're just interested in these issues related to data. So I'll stop there. We can take a few questions if you like, and move on.
There's a couple questions. I don't know if you see them, Bill. I'll read them until you find them. Adam Brickman had a question with respect to acquisition protocols. I think many centers interface or synergize with other studies or other clinical services that might impose restrictions on acquisition protocols. How do we resolve the differences from what we can do and what is expected? I assume there's no choice with the supplement, but data acquisition in general.
Yeah, so this comes down to, who's paying, to be pretty crude about it. And I think if NIA is funding the data acquisition through the ADRC program, and they're going to pay for the scans, I think they're going to, I can't speak for them, but I guess I can't speak for them. This is my understanding, that they want the data to be mergeable. But I think you'll find that our requests are not really that burdensome. And I think it's a very good question. It's a very important question. But I think you'll find that for PET, we're not probably asking you to do anything that you wouldn't normally do anyway. And for MRI, that's also probably true. And even if it's not true, what we're probably going to ask you to do for MRI is pretty limited. And I don't think most centers are really going to have problems with this. But, we'll have to, once you see the protocols, we can revisit that question.
Dean Coyne said, thanks for the great initiative. The supplement is a one-year supplement. That is correct. Are there efforts from this initiative or future plan that could help cope with harmonization and longitudinal follow-up when there are scanner upgrades, even when there are changes in scanner vendors?
So changes in vendors and upgrades have been a source of great interest, research, and consternation in ADNI. I know that both the PET and MR cores have spent a fair amount of effort dealing with this, and all I can say is we'll keep it on the list. I can't promise you we'll solve the problems, but we recognize that it's a problem.
Victor Henderson, will there be procedures to de-identify scans, specifically algorithms to prevent facial reconstructions from images?
Yeah, so that's another issue we're working on in ADNI. The short answer is almost certainly there will be. Precisely how we're going to do it, we don't know. I think there's a lot we could talk about here. Again, we've been concerned about this in ADNI now for a while, and we're going to probably end up adopting very similar methods to what ADNI does. So we still have to work these out.
Trey Hedden would like to know, for the single scanner, is there a PET MR combo scanner consistent with scan guidelines, and will there be needed adjustments to the protocol?
Yeah, so as you probably know, many imaging programs have excluded PET MR scanners. We decide we're gonna be brave, and we're gonna include them. So if you are planning to do your work on a PET MR system, that will be okay. We still have to work out some of the issues, and I think the biggest issue, of course, is what algorithm you're using for attenuation correction. We haven't gotten to that level yet, but we have decided that PET MR systems will be okay.
Bill, since you're working with so many different scanners and even PET-MR, why hold centers to doing imaging on a single scanner? I feel like that could be a limitation for subject scheduling and so forth.
Well, it's just a matter of making data compatible, and the more scanners we have, the bigger problem it's going to be. I guess we can revisit that, but this is what we do in ADNI now, and I guess, you know, I mean, one of the biggest problems with all this is longitudinal scans. You know, if you move across scanners in a longitudinal protocol, and of course that's not necessarily what we're talking about now, that's a big headache. So I think that's probably the key reason we need to do this.
Some of the other questions are specifically about the protocol, so I'm going to hold
Yeah,
those
I think we can
until you guys actually present what the protocols are. But we're just going to go to Beth right now, and she's going to give you the summary of the survey that you took a few months ago and go over those for you.
Okay, thank you. Yeah, next slide. Elizabeth, I think I did animations by habit, so you can just kind of pull them. Yeah, pull them all up. This should be the only slide. So, so I'll go through this quickly. This is posted, so you guys can download this and kind of look at it more details. But essentially, we got responses from 23 centers. The majority of these the responders were from imaging cores or biomarker cores doing imaging. So there probably is a little bit of a bias in these responses. We're blinded to the identity, NIA is as well. And the overall goal of the survey was to really assess interest in scan services and also harmonize prospective data collection. And we're focusing on MRI and PET. Next slide. So question one was really just asking about enthusiasm and interest in scan services, what Bill essentially just kind of proposed from the aims of their grants. Almost all sites indicated that they were interested in data sharing. So sharing of actual extracted data, sharing of data to qualified investigators, and also linking this data to other ADRC participants. There was less interest in services that are more related to kind of advice and guidance and web-based resources. But again, I think this might reflect a bias in the fact that people that responded are already involved with these established imaging programs. Next slide. So this question, I put a lot of text on this slide. And now that we've talked about this question quite a bit, and there are certainly a lot of ambiguities in the way that this was phrased, but we essentially wanted to kind of get a response related to flexibility on what prospective imaging might look like, specifically with what we're calling the core MRI protocols, which are the T1 weighted, the FLAIR, and the T2 star. And we kind of proposed two broad approaches. One is an exact version where there would be no tolerance for variation with the protocols that are pitched. And then the other option was what we called close enough, with the idea that this would allow more flexibility and not be as disruptive to ongoing studies. And pretty much everybody wanted the more flexible option. The top concerns that were raised is that without this flexibility, there would be limited kind of compliance and it would limit the amount of data that's collected. There was also a lot of concerns about ongoing studies and the funding mechanisms for these studies. But again, we're going to talk a lot about this, I think especially in Cliff's presentation, about what these two pathways actually mean, because it is quite ambiguous as stated in the survey. Next slide. So then we went through each proposed sequence and just got a sense of, what is actually already being collected, and then willingness regarding for scans that are not being collected, are the sites actually willing to do so? Next slide. So here are these three core MRI protocols I mentioned, so the T1 weighted, the FLAIR, and the T2 star. And for the most part, in terms of the sites that responded, everybody's collecting these sequences already. For the few that are not collecting these sequences, all the sites indicated they would be willing to collect this data. Next slide. Our optional sequences had a little more heterogeneity. So one optional sequence we assessed was diffusion MRI. Actually, in this case, most sites are collecting this, and those that aren't are willing to do so. Next slide. We assessed this kind of high resolution hippocampal T2 sequence. Less than half the sites are currently collecting it, but those that are not collecting it, many of them are willing. And then there was a handful of sites that indicated they would not be willing to collect this scan as stated. Next slide. And then resting state MRI was somewhat similar. So a little over half the sites are currently collecting this. Those that are not indicated willingness, and there was a few sites that indicated they would not be willing. Next slide. So here are the two PET protocols. There's a typo in the amyloid box. There's supposed to be ligands listed there. But we essentially listed kind of the main ligands that are being used and the corresponding time windows that are known to be best for those ligands. And for the most part, all the sites that responded are already collecting some version of this. And the few that are not collecting, all of them are willing to collect both amyloid and tau PET. Next slide. So then we asked, if you did have funding for this type of prospective image collecting, how many clinical core participants would you actually scan per year? And then so here, the mean response for MRI was about 100 subjects per year. And then for amyloid and tau PET, it was slightly lower. So it was right in the 50 to 100 category. So that could be interpreted as about 75 participants per year per site for both amyloid and tau. Next slide. Then we asked about participant groups. So are there certain types of participants that you would prioritize for this sort of harmonized scanning? And this was pretty consistent across MRI PET and MRI and the PET scans, but essentially most sites indicated that they would be most interested in scanning MCI, older controls, and Alzheimer's disease. And then there's a fair amount of heterogeneity across the other diagnostic categories, which makes sense that this reflects the heterogeneity across the ADRCs and the different focuses. Next slide. It was pretty much the same pattern for amyloid PET, again, emphasizing MCI, older controls, and AD. Next slide. And the same for tau PET. Okay, next slide. And then we asked kind of beyond diagnostic group, are there any other kind of criteria that you might prioritize for these prospective scans? And here, again, there actually wasn't really any kind of clear winners. The highest categories at 83% were those with longitudinal clinical data, and then at 74% of respondents was those that are newly enrolled participants into their ADRC. Next slide. And then finally, so after kind of assessing more of the scan-related topics, the Imaging Core Steering Committee was interested in understanding the variability and cost, because we definitely recognize there's a high cost to imaging, especially with PET. So then we asked for feedback on what sites are currently budgeting for these different types of scans. Next slide. So with PET, you have to consider both the cost of scan time and also of the ligand. So we broke it into those categories. So for PET scan time, there is a huge amount of variability. So, and I kind of just put the raw data up here because there's different ways of counting, of assessing cost, but overall, the range was between $590 to almost $3,000 for just scan time alone across sites. Next slide. And then here's for amyloid PET ligand cost. So I listed this kind of according to ligand, but in general, it was pretty surprising how big the swing was. So for PIB, for instance, their doses are being charged anywhere from $700 to $2,600 a dose. For florbetaben, it's $1,200 all the way up to $3,300, and florbetapir, it's $1,600 all the way to $5,000 per dose. Next slide. And a similar heterogeneity exists for tau PET. There's slightly fewer data points here, but the range was anywhere from $200 to $4,000 per dose for tau PET. Next slide. And then MRI, as expected, that was a little more straightforward. So most sites are being charged under $1,000 for scan time, and there's a couple sites that are just above that. Next slide. So to summarize, so yeah, we had a high response rate. 23 out of the 32 sites gave us feedback. There definitely seems to be consistent interest for what I'm calling ADNI-like services for data leveraging and access, so providing kind of data to the community. It seems that the core protocols, the T1 weighted, the T2 star, and the FLAIR, for the most part, are already in place with respect to the 23 sites that responded to the survey. There's a consistent enthusiasm for amyloid and tau PET. And then it does seem like there's quite a high potential for this prospective data collection, with 100 subjects per site per year for MRI, and then about 75 for PET. And then so next steps, so based kind of reacting to the results of this survey, as I mentioned, there's definitely these ambiguities surrounding what counts as close enough for MRI. And I know that Cliff will be discussing this. It was also unclear with respect to scan services, if this will be something for just the core and or optional MRI sequences, and how that interfaces with the degree to harm of standardization for all these different kind of protocols. It was quite interesting to see the high variability in ligand costs for PET across sites. And I do think that this brings up, opportunities to discuss, negotiation, like if there's ways for us as a larger group to make this more consistent across sites. This somewhat related to this, there's been some recent issues with distribution for AB 1451, but we have been kind of brainstorming on whether or not it would make sense to try to have coordinated efforts regarding radiochemistry production and distribution. I think it also will be useful to discuss priorities regarding who to scan and the scientific questions that can be addressed with this kind of imaging. It does seem like there's a lot of heterogeneity and interest in terms of the priorities. And importantly, we want to emphasize that this is the beginning of this conversation. We want to continue discussion and ensure that all the presentations today are clear. So I'll also try to promote Slack in an attempt to have some real-time feedback and communication. So thank you.
I didn't get any new questions based off of your presentation. We do have a couple minutes. If someone has a question and want to raise their hand, I will unmute you. But if not, we could, ooh, look at that. All right. Christina, go ahead.
Well, it's Jim using Christina's. I had to borrow. So I think this is awesome. It's fantastic. I think that Cliff is probably going to get into the close enough discussion, but since there was some time to talk about close enough versus right on is always best if possible. And most people don't really care that much about at least their 3D T1. And, Andersdale and I had worked, I think Anders is on the call too, but I had worked a lot with vendors to try to get them to just go ahead and put the latest ADNI protocol into their protocol selections on the machine itself. I think that would go a long ways to making it very turnkey to getting the exact protocol harmonized.
Thanks, Jim.
Hi, this is Jeff. I just have a general question. This is actually to use Cerise in the program. How many additional PET scan subjects do you anticipate being added to the overall NAC database through this supplement program?
Through the supplement program, we're thinking 300 or so.
Thank you.
Bill, if you're ready, you can go.
Okay, sure. So, all right. So again, for the PET protocol, it really is largely based on ADNI, though there are some distinct differences. So I'm just going to go through what we are going to do for scanner qualification, acquisition, data uploads, quality control, pre-processing, and data analysis. So the next slide. So if a scanner is qualified for ADNI, it doesn't have to be requalified. If it is not qualified for ADNI, it will have to be qualified following the way we do it for ADNI, which is basically imaging a Hoffman brain phantom, which we would supply with F18 radioactivity of some sort in it. And you have to do that twice, 2 days in a row or on 2 separate days. And that would be uploaded, and that would be the essentially done then, unless the scanner changed. And as we said, again, I guess we can discuss this. There's been some concern about this already, but we feel that a single scanner is going to be best. I mean, in a way, this is a program for the ADRCs, and it's kind of your call. I mean, the problem is the more scanners, the more noise, and that's kind of the trade-off. So the next one. So these are the actual protocols. So I guess I'll just go through them. I mean, I think people, this is based on what people are doing. It's based on best practices, but people are following the best practices. So for PIB, we're talking about 40 to 60 or 50 to 70 minute frames after a 10 to 15 millicurie injection. For florbetapir, 10 millicurie injection, and you see the frames there. Florbetapin, 8 millicurie injection, and 90 to 110. So that's for amyloid imaging. By the way, I think we will have a way of sort of relaxing some of the dose injections. Again, this is up to your local IRBs. So if you want to or are approved to inject more, we're not going to say you can't. There's no reason to do that. And if you're injecting less and getting good quality scans, we can probably be flexible on that, I think. So for flortaucipir, it's 10 millicuries and 80 to 100 minutes. For the Merck compound, it's 5 to 10 minutes and, I'm sorry, 5 to 10 millicuries and 70 to 90. There's some debate about this now, so we probably will accept 90 to 110 as well. For the Piramal compound, there's actually 5 millicurie injection and 2 protocols. If you're doing Alzheimer's disease, it's 45 to 75 minute frames. If you're Gil Rabinovich, you can do a 0 to 60 or 30 to 60 minute frames. I'm just joking. Gil wants to study FTLDs, and there is definitely a different temporal dynamics for that investigation. And we realize this isn't all the tau tracers out there. These are the ones that most centers seem to be doing, and there'll be new recommendations as new tau tracers come online. And then there's the FDG recommendation if people are potentially doing that. So next slide. Okay, so, again, back to the issue of innovation. If a center wants to collect some kind of novel frames or some kinds of dynamic data, obviously they can do that. It's just that if they want to get quote unquote credit for the scan, they have to upload the standard frames. And what's going to happen is if a scan doesn't comply with the protocol, we're not going to deal with it, basically. We're not going to QC it. We're not going to curate it, and it won't be counted towards the number of scans your center does. It'll probably go somewhere, and I mean, it's your scan. You can do what you want with it, but it's not going to be in the scan database. And again, as new tracers come on, we're going to try to do this by consensus and, of course, by data. I mean, what's the best time frame to use? Next slide. So image data are going to be managed by NAC and LONI together. As I said, we don't quite know what that interface is going to look like, but hopefully it will be invisible to you. It'll all go through an interface that we hope looks very much like NAC. And when you upload the scans, there's going to have to be something that we've called an information sheet that lists acquisition conditions and parameters. So someone is going to have to track that data, like things like injected dose and so forth, are tracked. Next slide. So you're going to use ADNI methods. Bob Kepi at Michigan is going to do the QC. And this includes checks like subject motion, field of view, looking at artifacts and checking things against the headers, and then pre-processing the images to get them into a standard orientation, standard voxel size, a standard intensity, and then a standard resolution. And so that is all very much what we do in ADNI, and all of these different image types will be available for download as we do in ADNI. Next slide. So the MRI story, so we've decided that we don't have to have an MRI to analyze PET data. We actually have already developed in Berkeley MRI-free processing pipelines that are actually remarkably good and quite yield results that are quite similar to the MR processing pipelines. We're not completely done with this, but I think that's where it stands. The reason for this is that ADRCs may be interested in recruiting participants that can't get MRIs. They have pacemakers or something else, and we can still accept their scans. If you are getting an MRI, we'd like to have it and have access to it, and we'd like it to be within a certain time frame of the PET. We haven't actually worked that out yet, but you don't have to get an MRI to upload a PET scan. And as I said, all the numerical data is going to go back to NAC, where they can be linked to the participant. You'll have amyloid status and so forth. And all the scans that are passing our QC and that have been harmonized will also go back to NAC. Next question. Next slide, I mean. Yeah, so again, I kind of went through this already. Maybe I'll just re-emphasize that you'll get regional SUVR data in a CSV type format, and that's available for anyone to do what they want with. We'll also provide an index of amyloid positivity for the amyloid scans, and for tau we'll provide some kind of aggregate summary ROIs based on Brock staging and the meta ROI that Cliff Jack came up with, and similar for FDG PET. So that's all going to be available in CSV files, and will be linked to participants on the NAC website. Next slide. That was it. Okay.
Bill, do you see the Q&A box?
I do.
There's like 6 questions that are open. If you could just spend a few minutes.
The first one's from Thomas Wisniewski. How long will the MR protocols take? I'll just tell you the required protocol is about 10 minutes, but Cliff will get into that in more detail. From Helena Chu, do the MR and PET need to be collected within a certain period of time? That's a good question. Honestly, we don't really have the answer to that. I think, I don't know at this point. We'll probably would like the MR to be collected in a reasonable time frame with the PET. This is from David Wolkin. I'm going to leave it to Cliff to answer this one. It's about the MR protocol, so I'm not going to get in hot water. This is from John Ringman. As the idea is to study the ATN framework, is the expectation to get amyloid, tau, MR in every participant? If you can't get all, you can't get none. That's up to you. We would, our only concerns is what are the quality data, and who you want to study and how you want to study them is up to you. And it's really up to you and NIA, because NIA has an interest in this, obviously, but we're not going to make a decision on who you scan or what scans you get on people. That's your call. Are there criteria for what participants can be sent through this pipeline? Again, up to you, not up to you and NIA. Do you prefer MR and PET to be conducted at the same time without a delay? Well, no. I mean, I think we would like to have, we have not actually stipulated this yet, and I don't know what the answer to this question is going to be, but obviously we'd like to have them within a reasonable time of one another. Is the MR-free PET analysis based on warping to standard space? If so, do you have age-specific templates? The answer is yes and yes. So we have age-specific templates, and we have and we're warping to standard space. And like I said, so far it works. I mean, if you take the data we've analyzed with our MR pipeline and our MR-free pipeline, is a remarkably high correlation. Next question has the word fund in it, so I won't. I'm going to let that one go to Cliff also. That's about the MR. I think that's it. Oh,
Tom Vajewski put another one in. Do you plan on expanding ATN to include specific vascular measures?
well, as you'll see, the MR protocol is going to require a flare sequence. So from that, one can get vascular measures. I don't think there's any agreement on how V should be categorized in the ATN quote unquote V schemata. So we can just do what we can do, which is to quantitate the flare images and other images, as you'll see, in the best way we can. And how that enters into phenotyping will that's not our ability to determine. Oh, here's a hard one. For ADRCs with 3 sites, should we be choosing one site or one scanner? Well, I mean, there are limits to what we can ask. I mean, if you have 3 separate sites in your ADRC and they're geographically split and participants don't routinely go from one to another, I don't really think we can ask you to use one scanner. That seems, honestly, I hadn't considered that and we haven't talked about it, but it doesn't seem very realistic to ask you to shuttle your participants around a wide geographic area just to keep them on one scanner.
Thanks for that feedback. I'm sure that the steering committee can get additional information.
Yeah, I mean, to be honest, a lot of these questions I hadn't anticipated. I don't think any of us had. I mean, we've only met like twice since we got funded, and even though we drew up our battle plans in the grant, they say your battle plans usually fall apart when you confront the enemy. Not that anyone's the enemy. It's just that we really hadn't thought through a lot of these things. So I think these are great points that we're going to have to think about some more.
We have about 5 minutes. And so if you want to raise your hand and ask questions, I'll unmute you. But then, Bill, there's another one that popped up in the question.
To be eligible for the supplement, can we propose to do tau PET and MR only without doing A beta PET? So again, that is between you and NIA. I'm sorry, maybe this is meant for you, Cerise. I don't know if you want to answer it.
Yeah, we would have to look through your plan, and so yes, the limit is 250K. So if you feel that your proposal is the best in which to spend that money, we'll review it. If you, I don't see any hands to ask a question live. I'll count to 7 before I decide. Okay, Cliff.
All right. Okay, so I am going to try to share my screen. Okay. There. I don't know if is that showing up?
No.
It's not showing up. Oh.
When you see your screen, you have to pick like that PowerPoint square and then hit share again.
Oh, screen 1. Okay. Is that showing up?
No.
Okay. Does that show up?
No. Does Elizabeth have a copy of your slides? Oh, there you go. It started.
Oh, okay.
Now you need to do slideshow.
Does that show up?
It just show up, and now you just need to do slideshow.
It should be slideshow, on slideshow, yeah?
No, right now it's on like the tile of every slide.
Okay, how about that?
Now you got it.
All right, okay. All right. Okay, so yeah, so Bill mentioned this, and I'll just reiterate it. because this is really how Bill and I both approach this whole project here, the SCAN project. And that is that our goal is to advance the priorities of the NIA and the ADCs, and we really view SCAN as a service to the NIA and the ADCs. One of the overarching objectives, as you've heard from several people now, is to introduce an element of harmonization or standardization into the ADC imaging programs overall, and to try to use approaches that are similar to what ADNI's done. So what I am going to do is first review, for people who may not be aware of it, how ADNI has approached this notion of harmonization, standardization, quality control, etc., and then indicate what we see as similarities and some differences between what has been done for ADNI and what we are proposing for SCAN. So, MR in clinical trials or an ADNI environment, ADNI is obviously designed to mimic or mirror a clinical trial environment. So the first thing that we did and would in ADNI and would do for any kind of clinical trial is a site survey and then develop a protocol. And we propose to do pretty much the same thing for SCAN here as was done for ADNI. So we would take a technical survey of the scanners at each site and pick the best one for the study. Now I'll come back to that in a few minutes because obviously that's only applicable for sites that are newly starting up. We would first create a generic non-platform specific protocol. What does that mean? It means that we select the sequence types in the protocol, the order that they occur in, and some parameters. Then we have to create a vendor and operating system specific protocol for each scanner in the study. So what does that mean? This is mainly for people who aren't all that familiar with MR. How MR works at sort of a detailed level. So this is what the generic ADNI3 generic protocol looks like. You can see the different imaging sequence names and the order in which they occur. There's 7 of them. But in order to operationalize this on any scanner, a number of parameters have to be specified. So this is just the parameter specification for one kind of imaging sequence, for one operating system, for one vendor. So this is a sagittal MP-RAGE. You can see in the columns here the different parameters that need to be specified, and then their values are seen in the adjacent column here. So there's just one sequence here, 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 here. 5 pages. So there's about 150 parameters that have to be specified exactly for each sequence. And if you have 7 sequences in the exam, then that's about 1,000 parameters that have to be specified in order to run a protocol in a harmonized way. So that's a lot to keep track of. And to do that manually is a guaranteed recipe for error. So how do we manage this? We manage this by sending and then having the site load the protocol onto their scanner via an e-file, electronic file, which will then load and save all these the protocol specific instructions. So the way we qualify a site then is to send and load the protocol e-file, scan a phantom or a human, and then we at Mayo would get a hold of the scan, the qualification scan, verify that the site executed the protocol correctly, and if the answer is yes, then the site is certified. At the participant exam level, and we propose to do pretty much the same thing for SCAN as we have been doing for ADNI here, the participant scanned, the data is sent to a central repository, pulled by Mayo, and then we do several things. One, we do automated quality control. So what are we looking for? We're looking for to verify that the correct scanner was used, the one scanner at that site that was certified for the study. The correct patient was scanned. You'd think it would always be the right patient, but not necessarily so. And that the protocol was correctly executed. This is done by using a software program written here to automatically correct or compare the parameters, all parameters in the for each sequence in the scan that's been done against the master. We then manually review the images in each exam, and we propose to do the same thing for scan as is done for ADNI. So we look for medical findings. For example, does the patient have a recent infarction? Is there a tumor present, hemorrhage, etc.? And if present, of course, we notify the site and the study. This does not mean, though, that local interpretation of MR scans is not needed. It is still needed because no central hub, if you will, or no central managing group can be responsible for local medical legal sorts of things. So a local MR interpretation is still needed. We then do visual quality control, where we look for scanner problems, technical scanning errors, etc. So what kind of things are we looking for? Well, here's an example. I don't know if you can see the cursor. Can you see it? I move it around.
Yeah.
Okay, good. Okay, so here's an example of a site that was scanning. We got this scan. You can see these dark areas here, dark area here. So this was a site that was scanning with a head coil that had some bad receiver elements in it. So we had to notify the site that they had a problem with their head coil, get the service engineer out there to fix it. Here's a site who scanned in the wrong acquisition plane. So if you look down here, the patient actually does not have a lesion projecting out of the clivus into the brainstem. This is just unsuppressed cerebral spinal fluid aliasing backwards into the brainstem in this participant who the site scanned with the wrong scanning plane. So this is something they had to correct. This is a obvious copying of the field of view prescription error. Again, this is something that had to be corrected. Here's a site that it accidentally turned off the on-scanner or online intensity normalization that is part of image reconstruction. Again, that's an error that had to be corrected. So those are the sort of things we look for scan quality control. And the plan for scan, the SCAN, the project, is that this front end sort of things, protocol creation, the site certification, quality control, everything up to image analysis would be done at Mayo as it's done for ADNI. Then the image analysis part, as Bill mentioned, would be split between Charlie, DeCarli's group, and our group. So how to operationalize the MR for scan? Like I said, many aspects of the ADNI operationalization seem like they're a good fit for scan, but some may be problematic. And Bill has already discussed one, and that's the notion of one certified scanner per site. Not per ADC, but per site. And really that, I think that's an important aspect of harmonization. If you have a participant who bounces around from scanner to scanner on successive exams, you're going to introduce a terrible amount of noise in the longitudinal trajectories of that participant's data. And that's something that does not exactly fall in line with the notion of data harmonization. A second thing is that for ADNI and for clinical trials, we try to pick the best scanner at each site to do the scans on. Now, to do the MR exams on. This seems like it's something that would be helpful for ADCs who are newly starting up imaging programs and who may not be all that familiar with different MR, the quality, the properties of different MR scanners. And this is something I think that we could help sites with. Established sites who have established longitudinal imaging on their cohorts are probably not going to want to switch scanners. The final thing I'll mention about operationalization concerns the protocol, and the questions that have come in and have already started honing in on this problem, and a lot of discussion, as Bill mentioned, has been focused on this issue of how to harmonize the MR protocol. So this is what the ADNI 3 protocol looks like. It's about 45 minutes of actual scan time, about an hour. The patient would have, or the participant would have to be scanned for roughly an hour to accomplish this. They'd have to lay on the table for about an hour. And you can see that if each one of these things was done and was required to be done in a standardized way for ADC participants, there would be no room at all for innovation at the site level. I mean, it's unreasonable. It would constitute an unreasonable burden to participants to ask them to do all of the stuff on this protocol and then other things that the site might want to do for their own innovative purposes. And so this brings up this notion of an inherent conflict between on one extreme requiring everyone to do exactly the same protocol. It's great for study-wide harmonization. At the other extreme, you have sort of free-form acquisitions where sites could do whatever they wanted. It's great for innovation, flexibility. It's also great for consistency at the site level, but it does nothing for data harmonization. So for example, a site might want to optimize, imagine two different sites. One site has the scanner you see on the left, which is a better scanner than the scanner that you see on the right. So the, the scanner on the right just has worse signal to noise properties than the scanner on the left, and there's nothing one can do if you hold time constant, acquisition time constant, there's nothing you can do to make the scanner on the right, the scan on the right, look like the scan on the left. The people who have the scanner on the left might want to trade off some of this better signal to noise for better improved spatial resolution, i.e. smaller voxels. And if you restricted the spatial resolution to a common denominator in the protocol, you'd be preventing a site like the one who might have a scanner like the one on the left from doing that sort of innovation. So we have this couple different options. At one extreme, no harmonization. At another extreme, rigid harmonization. Neither of those really seem like they fit the needs of the NIA or the ADCs very well. But we've discussed this notion of a compromise, and this is what this compromise solution is what we're proposing for SCAN. And that is that a site could choose between two options. So option one would be that the site would only have to do two ADNI sequences. So the site would have to do the ADNI sequences exactly as they're specified for ADNI, and this would give good harmonization across sites. But this will only be for the T1 and the flare. Each of these takes about 5 minutes, and so this would mean that the first, and I emphasize first, we don't want these two stuck at the end of a 50-minute exam when the patient's tired and moving around. But the first 10 minutes of each exam belongs to the NIA, but the remainder of the exam belongs to the site. So the site could do whatever else they want in the remaining X minutes of their exam time. And this would allow the site to, one, retain all or most of the sequences that they've been using for existing longitudinal studies so that they can be consistent longitudinally with their data. And also, it would allow this site to innovate. And SCAN would only collect and analyze the T1 and the flare because these then would be the two sequences that would be standardized, that would conform to this notion of standards. Alternatively, a site could do more than just these two sequences. They would do the T1 and the flare, but then they could add additional standardized ADNI sequences if they wish to do so. And in this case, SCAN would collect and analyze all the sequences, all the standardized sequences that the site would do. And we envision that this might be particularly useful for sites that don't now have a formal ADC imaging program, but that want some help getting started. And this would, again, be a service that SCAN would be providing to these sites to get things up and going. So schematically, then this is how it would look like. Option one, option one for the SCAN protocol would just look like this. You would do the 3D T1, the standardized ADNI 3D T1, the standardized ADNI 3D flare, and then the rest of the exam would be whatever you want. ADNI would collect these two, first two, and ignore the rest of the exam, which was done in a non-standardized way. Option two would look like this. you would do the 3D T1, then the 3D flare, but then you could do other sequences here using ADNI standard methods, which we would again standardize as best we can across sites. This is a little bit vague because if you recall the survey that Beth showed, some of these there just did not seem to be a whole lot of interest in. There didn't seem to be a lot of interest on the part of many sites in task-free fMRI or the coronal high-resolution T2, both of which are part of the ADNI protocol. And if it turns out that there's not much interest on the part of sites, we may just drop these from consideration. Also, the T2 star GRE gets a little complicated. I won't go into details on that, but there are other options other than just doing a standard single echo long echo time gradient echo scan for that. I want to finish up here by just emphasizing this idea that as much as we try to standardize, there are limits to harmonization. So here are 3 different people scanned on different scanners. They're different people, but they used the exact same protocol. So same spatial resolution, the same TRTE, et cetera, to give nominally the same contrast properties. So this is as standardized as we can make it using the product sequences on these scanners. However, I don't know if this is all that visible but I think you can see on this Siemens biograph. So this is the MR scanner for a PET MR, Siemens PET MR. And you can see that the speckle in this scan, and compared to the signal to noise properties in this image right over here, it's very clear that the signal to noise in this scanner is a lot better than the signal to noise in this scanner. And there's nothing that can be done in terms of acquisition to make this scanner give equivalent signal to noise to this scanner. This is just a better scanner. Similarly, you can see that the contrast properties here don't look exactly the same as the contrast properties here. The filters that are applied at the time the data is reconstructed for GE systems are not the same as the filters that are applied on Siemens systems. These are properties of the data reconstruction methods that are buried very deep in these 2 different vendors' approaches, and there's nothing that really can be done to undo that. Similarly, for flare scans, nominally it is identical resolution, nominally the same parameters, but you see that the signal to noise is worse here compared to here. The contrast is not the same for this as it is for this. There's very little that can be done to correct this on the acquisition side. The question you might ask is, does this really matter? Does this level of residual non-uniformity or non-standardization, after we've done everything we can to try to standardize it, does it really matter? So here's an illustration that it does matter somewhat, not a huge amount, but it does. I don't know how effective this illustration is going to be, but these are 2 different scans, one done on a GE scanner, one done on a Siemens scanner here at Mayo. Same participant, the scans were done about an hour or 2 apart, and then pumped through exactly the same image processing pipeline for gray matter segmentation. So everything was constant except the scanner. The resolution was changed a little bit, slightly better for Siemens, so that is a wild card. But the main thing is that the vendors were different. And this is what the gray matter cortical segmentation looks like on the Siemens system, and I'd draw your attention to the area of the brain that's here circled in green. It's what it looks like with the Siemens scan. It's what it looks like with the GE scan. So I'll just toggle back and forth. And I think you can see pretty clearly that it's the same patient, it's the same image processing algorithm, but a different scanner. This is just one slice, but you can see that the notion of what constitutes the gray matter ribbon is different. And this is just one slice out of many, and this sort of discrepancy exists across the brain. there's really not a lot that can be done acquisition-wise to correct this problem. However, a lot of work has been and is being done to try to wring out data heterogeneity in large data sets after the fact. And this is the last slide I have. And I would just, this is a lecture or a brief review recently given to the ADNI-MR core by Paul Thompson at USC. And for people who are, there are a variety of approaches that can be taken after data is acquired, including AI-based training adversarial networks to try to wring out scanner-specific effects or site-specific effects. And for people who are interested in looking at this, this is a link to the YouTube video of Paul's presentation. It's short, it's very informative, but SCAN is going to, like ADNI, be looking into these sorts of techniques. A big part of what we're going to do in terms of data processing is going to focus on this sort of thing. So that's my last slide, and I'll stop there.
Thanks, Cliff. We have about 10 questions in the queue. So are you able to see them, or do you need me to read them?
Yeah, so David, first question I see is from David Wilk. Yeah, if the end product of these scans are, how open are you to allowing small variations in MR parameters? IER Center collects 0.8 millimeter isotropic T1s, so does ours. That's their, if it could be shown, yes. So that's really the point of this whole, David and others who are interested in this, that's the whole point of this notion of, we have two standard ADNI acquisitions, the T1 and the flare. Then if you want to do a 0.8 by 0.8 by 0.8 T1, that's fine. That becomes the second 50 minutes or whatever of your exam. It gets added on, but doesn't replace the standardized 1 millimeter cubed ADNI acquisition or scan acquisition. I hope that answers the question. And we think that by only requiring two sequences in order to quote unquote qualify for ADC compliance, that leaves plenty of time in a typical exam to do other things that you want to do. In this case, what you're saying is do a 0.8 millimeter cubed T1. Okay, are the funds to be applied only to the standard scan protocol sequences, i.e. if we add additional MR sequences, is that portion of the scan cost to be covered by other means? So I am not sure how to answer that question. And
yeah, the funding questions are for NIA. We can individually to folks about those.
All right. I mean, so I'm not going to say anything about funding, but just for your own, for your own edification, MR, most places, you buy a time slot for MR. You do not buy time by the minute. You buy it by the whatever 40-minute slot or 1 hour slot or whatever. And once you've bought the slot, you can do whatever you want as a user with that time. So the idea of paying for each additional sequence doesn't not match up very well with how MR charges are done in most places.
But also, if they're doing the standard scan protocol, I think you discussed earlier about adding additional MR in sequences, and that's allowable to do.
Sure. Yeah.
That you have allotted.
Yeah. So scan will pay for an exam, or not scan. The NIA will pay for an exam to qualify for ADC or /scan/NIA compliance. All you have to do is the first two, T1 and flare, and then after that you can do whatever you want. So the NIA has paid for that exam time, and you can do do the first 10 minutes. First 10 minutes belongs to the NIA. The rest of X time of the slot belongs to you. It's a good deal. Is there a requirement or recommendation for how soon the MRI or PET needs to be done after the last clinical psychometric evaluation? I don't think that's something that SCAN, as Bill mentioned, that's not really something that SCAN is going to get into, dictating or mandating or anything like that. Really, that's up that's the sort of operations of how your ADRC is set up to work. Obviously, the minimum time gap between these various data sources is best, because then the notion that they all point to the same point in time is most valid. But we're not going to get into specifying time gaps. Let's see. For SCAN, would you be required to collect isotropic flare if current resolution is higher than blah, 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 blah. Okay, so again, we have a standard. We have an ADNI standard for the flare. But if you in addition to that, you want to do something different, if you have a really high, good scanner, then you want to trade off some of that signal to noise for better spatial resolution, by all means do it. But that's a separate acquisition that would not count as the EDC acquisition. If an ADRC scanner has not yet been ADNI certified, will there be an impediment to applying? I don't think so. I mean, it's not as if there's some question. It's not as if though there's going to be a question about your being able to certify a scanner. At some sites, it takes longer than others for certification for a variety of reasons. But we eventually will be able to certify any scanner. So I wouldn't worry about that. If we are, I think this is Adam Brickman's original question. If we're collecting core scans, T1 flare from other protocols that do not conform exactly to the SCAN protocol, will SCAN still accept the data, i.e. will we get credit? The answer is no. We want you to do the T1 and the flare from the ADNI protocol, but you can do your own T1 flare that you've been doing in your participants for however long, in addition to the ADNI T1 and flare. We wouldn't collect it because it's not standardized. But again, we don't think that 10 minutes of acquisition using the specific ADNI standards is going to be a significant impediment to innovation. Why, next, Ranjan, why would you not include T2 star GRE and a diffusion sequence among the mandatory sequences? Should we not always try to identify microhemorrhages in vascular disease and recent infarcts? Well, the more that you include as the mandatory sequences, so it's unreasonable to ask a participant to lay on the table for more than an hour. The more you include as mandatory sequences, the less time there is for sites to innovate. So there's a trade-off, right? We could say, so there has to be a trade-off. And so we, both Bill and I, and also the people on the imaging core, think that the two sequences that have the, let's say, the biggest diagnostic impact are going to be the T1 and the flare. And we had to come up with, we had to trade this off. We have to we have to minimize the required sequences to a sufficient degree that it does allow sites to basically pack in a pretty full other set of sequences that they want to do to maintain their own longitudinal consistency.
Could I just add something to that?
Sure.
I think this is a great example of how the centers can drive this whole thing, right? So, for example, I mean, what we're trying to do in SCAN is facilitate sharing of images. If the centers as a group, decide that they want to prioritize a more in-depth measurement of vascular pathophysiology with T2 star or diffusion imaging, and there's an upsurge of interest in doing that, then the centers should get organized and do it. They should get organized through the imaging steering committee, and they should say, let's get together, how many centers, 5, 10, whatever, and do the diffusion and T2 star sequences. And they're there for you, you can use them, but I think it should be driven by the science has to be driven by what the ADRCs want to do. If your center wants to collect those data, that's great. If you want to organize 10 of the centers to do that, that's even better. And so you have I think the centers have the flexibility to do this sort of thing, and it's really up to the centers.
Thanks, Bill.
Yeah. All right, so Sam Lockhart, sorry if I missed it, any instructions for MR head coil? So again, we will do an MR survey at your site. And part of the survey includes, it's a very detailed technical survey. It includes, of course, what available head coils you have. And, we can tell you what the best head coil is to use. But we're not going to, I don't think we're going to mandate that you have to use a certain head coil because it may be very difficult to access that coil or whatever. I mean, these are at your site. These are sort of site-specific things. What we want, of course, is always the best we want the best equipment at each site for scan and for the ADC protocol. So if you have an old-fashioned 8-element coil, and then you also have a nice hot 32 or 64 channel coil, of course we'll tell you to use the 32 or 64 channel coil. Okay, Bill, I think the next one's for you. Pet.
Will there be rules, restrictions on what participants can do during PET tracer uptake? Not for amyloid or tau. If we do FDG, we might talk about that, but even that, I'm not a firm believer that really is that important. But for FDG, it certainly has some importance. I think for the amyloid and tau ligands, unless you're collecting, no, I mean, for the amyloid and tau ligands, it doesn't make a difference.
Okay, next, Lon Schneider. Okay, so right, I think that this is an earlier cohort is scanned on scanner A. I'll read it. It's a long question. An earlier cohort is scanned on scanner A and continues to be followed for 10 to 15 years. A newer cohort was started on scanner B and has been followed for about 1 to 3 years. It seems like you would be asking the site to choose between the cohorts for participation in scan. It would seem preferable to divide the site into A and B. This is also analogous to ADCs with several sites to make this more complex. The ADNI cohort of say 35 might be followed on scanner A, but the site might be more partial to scanner B. Comments. So what I would say is, it's complicated, but what I would say is at this site, it does make sense for scan to qualify 2 scanners, A and B, and the cohort that you've had on scanner A, keep it on A. The scanner cohort that you have on scanner B, keep it on scanner B. But we do keep track of the scanner. Each, what scanner each exam is done. There's a serial number that's embedded in the DACOM header, so it's not a secret. We know what's going on, and we would say keep the people who've been scanned in your cohort on scanner A on that scanner, and likewise on scanner B. Will the data be shared immediately like standard ADNI data? That's a good question. I don't know that we have so I could tell you what happens for the MR part of ADNI, and Bill can comment about PET. But what we do in the MR part of ADNI, there are a number of different investigators who've been assigned different measures to make, and we actually have quarterly deliverables. So we keep track of, people are required to upload their particular data analysis stream on a quarterly schedule. And we track that every quarter. And I don't know if we haven't really discussed this, whether we want to keep the same sort of thing for scan, if we want to try real time.
Yeah, I mean, I can sort of see where this is coming from. So, you know, in ADNI, for PET, I mean, it's fairly similar. We do pretty almost real-time QC and harmonization. So Bob Kepi has the images QC'd and checked and processed and back up on the database pretty soon after they've been acquired. But we, frankly, it's just not labor. It's just not a good use of our time to run our pipeline every night when there's 1 or 2 new scanners. So we tend to batch them. But I think if the ADRCs, for example, want a faster turnaround for some kind of reason, I think this is something we could probably accommodate, which is to say, rather than batching our PET analyses every 3 to 4 months, as we currently do for ADNI, we could probably do them faster. But I think we have to have some back and forth with the ADRCs about why they want that and what exactly they want, because we certainly can't provide a diagnostic radiology type of environment where you get the scanner results back 24 hours later or something like that.
Yeah. So the next question by Judy Heidebrink. I think, you know, Bill addressed this earlier, and that is, so scan, when it will begin, I'm not sure, but sometime. And whoops, it already was lost. Could you go back one?
It was about whether it's prospective data, and the answer is yes. It's only, so what we've been calling in scan legacy data, that is data your center might have collected previously, we are not going to deal with that.
Yeah, and so the second half of that question was, does legacy data that's quote unquote close enough count? And it doesn't matter because legacy data doesn't count. So whether it's close enough or not would be not relevant. So next is, you know, Bill mentioned this. So NAC and Lonnie ADNI have very different image uploading interfaces with a different philosophy on when, how de-identification of the scan takes place. Will there be a survey of the site into which this is preferred? And what best, okay. So the answer here, Bill again alluded to, and we think that what's going to happen is that scan is going to use the Lonnie interface. So the look and feel of data upload and de-identification from the site's perspective would be what you experience with Lonnie right now. Okay, this is another complicated question. To follow up on Lon's question, an old scanner may be ADNI qualified but heavily overscheduled, while an equal same vendor or model more flexible scanner is available and used by some ADRC collaborators for a number of reasons. We would be willing to get it scan qualified and further ensure that subjects scanned longitudinally would be kept in the same scanner as their original. This would help us significantly because we have a MOU with users that ensures that they will follow ADNI protocol for T1 scans and soon with FLAIR and return the results to the ADRC. This gives us several free MRs that are ADNI compliant. So I'm not sure what the question is, but you know, in terms of from the MR side, I think that we are going to want to requalify every scanner for scan. And the reason is that, if you already have an ADNI qualified scanner and you are a site that, you're probably, I'm going to guess, are going to be a site that's going to do option 1. That is, you're going to strip out the T1 and the FLAIR, and you're going to add a bunch of sequences that you already want to do to the protocol. So you'd be doing a different protocol. You'd be doing a protocol that's actually different from ADNI for scan. And so we would want to qualify that. It'll be very simple. You just scan a phantom and send it through. So it
Cliff
would not,
and Bill, sorry, were you done?
okay, I can be done.
Okay. Adam Brickman had a question. He's going to ask it.
Sorry, Cliff. Hi, it's Adam. I don't mean to perseverate on this thought, but I'm getting a little bit confused between what sort of scan, whether scan just applies to the data that are being collected as part of these supplements, or whether it's applied to any data that's collected in the ADRC. And I ask, maybe this is unique to Columbia, but we try very hard to leverage other resources. For example, the clinical radiology department has agreed to do a harmonized or standardized MRI protocol for any clinical patient who ultimately might get enrolled in the ADRC. Then we can grab their clinical MRI data, but they won't use a 3D flare. They insist on 2D flare for whatever reason. So there's still standardized data. We don't have the ability, because they're required initially for clinical reasons, to add an additional sequence, but they're not exactly in line with what you're proposing in SCAN. So we would still use that data, but it sounds like we would not be able to upload that for databasing in NAC and central processing. Is that correct?
So one, that's correct, because you wouldn't be doing the flare according to the protocol. But two, maybe you want to take this offline.
Yeah.
But I mean, if you're getting scans for clinical purposes, then presumably those scans are being paid for via a clinical cost center, and using the whole NIA reimbursement scheme for scans that have already been paid via a clinical reimbursement cost center seems like double dipping.
No,
so I don't think you want to get into this.
No, we're not doing that. We're just getting permission. We're just getting permission to use clinical data that have been collected. We're not paying for any of it through NIH dollars.
I see. Okay.
I mean, I think that brings up the general issue. Again, we know from all the surveys that we did that lots of ADRCs are collecting a lot of imaging data in very different protocols, and these are, up until recently, these have largely been funded either by clinical dollars or separate R01s or separate grants of other types. And I mean, I don't think that the NIA is, I mean, I think what we're talking about now is what the NIA is funding through this supplement. How that is rolled out over time, I think, is something that, Cerise, Nina, and NIA folks are going to have to weigh in on in terms of, in general, how does this relate to all NIA resources? And I think in general, our thinking, based on discussions that we've all had openly, is that when NIA resources are used for something, they ought to follow the SCAN protocol. When other resources are used, then it's difficult to insist that you do it our way.
We can go back to the box. James Becker had a question.
Yeah, so this is again about a single scanner per site. So so our site is adding, I'm just reading, our site is adding a third Prisma to meet the current scanning needs. However, with COVID virus safety rules, it basically, what he's saying is it'll be hard to scan ADC subjects on the same scanner. Our safety rule, we reduced from 10 scans per day to 5 scans per day per scanner. And what he's asking is, can you use different scanners? I think this is something we're going to have to evaluate. We're going to have to think about. Obviously, there's some pushback from the sites about this notion of single scanner. But the reality is, if you have a person hopping from scanner to scanner, and those are different operating systems, and particularly if they're different vendors, you're going to introduce a tremendous amount of random noise in that data stream. And while it may be convenient, that's not very good data. So I think this is something that we're going to have to, it's clearly an issue with the ADC PIs. It's come up a number of times. But it's something we're going to have to discuss. And I don't think I have a clear answer right now.
With this, Sterling, I think that if we can try to be flexible there, if there's a strategy we can use so that the subjects are scanned on the same scanner and both are qualified, it seems like that would help a lot of the centers.
Yeah, if it's a couple scanners. On the other hand, if a site was to say, well, we'd like to qualify 15 scanners,
Yeah.
there's a problem. So that's why I think we need to think about how exactly this would be operationalized. Sure.
Hi, this is Annie Cohen. I just wanted to clarify real quickly. I think that Jim's question, and again, not to perseverate on an issue, but if our site has 3 Prismas with the same, or soon to be 3 Prismas, same operating system, 2 of them are already ADNI qualified, and we always commit to keeping the individual participant on the same scanner, but we have multiple scanners so that we can get the throughput we need. That's sort of different than jumping from scanners with different operating systems and different vendors, right? That's sort of, I think, what we're trying to clarify here.
So the answer is yes. So a couple of things. Number one, the reality is if an identical scanner model with an identical head coil and identical operating system, that's not going to make any difference in terms of, that's not going to introduce noise into the data stream if people move around from scanner to scanner. It's changes in vendor, it's changes in receiver coil, major changes in operating system. Those are the kind of things that introduce noise. So I think that, I think, we will be flexible on this, but like I said, there has to be some reasonable limit. I mean, we can't qualify 15 scanners at a site, just on a whim. I mean, so we'll for sure discuss this and come back with recommendations.
Yeah, I think we're going to have some offline discussions about the question of single scanners. Clearly, it was a bigger issue for the ADRCs than we thought it would be.
Right.
So we'll get back to you guys on this. I understand it's an issue. So I think the next one, so this is about that question of how quickly can we turn things around. The motivation is to provide ATN classification to clinical core and collaborators. So I understood that when I answered the question. I mean, I understand that ADRCs want the results fairly quickly. And like I said, I think we can try to do it faster than once every 3 to 4 months. But I don't think you can expect what you would get from a radiology department. I just don't think my lab is geared up to do that. We can see how quickly we can do it. For example, it might be reasonable to batch things weekly. And if we can do it overnight, we'll do it, but I wouldn't count on that. So the short answer is I just don't know how fast we can turn these things around. We were planning on every 3 to 4 months, but if there's a real need, we will try to do it faster. We'll just see what we can do.
Let me just, amplify that. So one of the steps that we do in our quality control here at Mayo is, we do these uploads in batches, and we, do a statistical analysis of each batch, looking for outliers and this sort of thing. And a formal statistical analysis that runs through our biostatistics department is not a real-time kind of operation. It's a batched, it's the kind of operation that, that really has to be batched. But I think it's good for quality control. So I'll leave it at that.
The next one, I guess, is the one from Trey. Weekly would be fine. Well, I'm not promising weekly. I know that's Jim Brewer disguised as Christina Gigliotti. So I know that's you, Jim. I'm not promising every week. We'll see what we can do. So there's one from Trey Hedden about if multiple A-beta ligands are available to a site, will data from multiple ones be accepted by scanners? Should all data from the site be done with one ligand? If one is preferred, is that only for the supplement or in general? No. So, you know, this is, again, I mean, an area where you're going to have to make that decision. We have listed the ligands for which that most sites are using, and actually, we didn't cover them all because we didn't have criteria up for the NVIDIA compound or for flutometamol, but any amyloid ligand is going to be fine. And if an ADRC wants to do more than one, that's okay. We'll convert them to centilloids. But it's still, you know, it's better for you to stick with one ligand as much as you can. And certainly, if you're doing a longitudinal study, you should stick with one ligand. But, you know, this is an issue for peer review. It's not an issue that SCAN is going to determine. If you can convince a peer reviewer that they'll to accept a longitudinal PET scan study where you use different amyloid imaging agents over time, go for it, you know, but I don't think it's a good idea. So this is from Ranjan. How would you be standardizing SUVR and centilloid calculations? Will they be done at the ADC as well as by SCAN? I'm not quite sure what you mean, but ADCs are free to do anything they want with the data. Those scans, you know, obviously you have the raw data, you'll have the processed harmonized data, you can do anything you want with them. We will process the data in our pipelines, and our pipelines have been certified, have been designed to with conversions into centilloids. So we'll give you the centilloids that come out of the SUVRs that we measure in our pipeline. And but if you know, I mean, you should probably use one or the other. Hopefully the answers won't be too different, but you know, you have the either option.
There are no more written questions, but Dave Walk just put one in. But if you want to raise your hand and speak a question, I'm open to unmuting you. So Dave Walk, sorry.
Yeah, so this is, can you clarify whether SCAN will accept subjects with partial ATN data, i.e. MR without PET or PET without MR, just a single PET modality, and for the case where someone has access to conforming 3D T1 MR, but not a conforming 3D flare, would SCAN accept just the 3D T1, which would be sufficient for assessing N, but not V? Okay, the first question's easy. The second one I'm going to let Cliff answer. The first one is, can you get just an MR without PET or PET without MR? Yes. The second part is, Cliff.
Yeah, so the second part is, you know, I guess the first thing I'd ask is, the conforming flare takes 5 minutes. It wouldn't be that tough to add. Whatever you're doing, whatever exam you want to do, it would not be that tough to add 5 minutes of acquisition time to you know, what you're doing, and then you could get the conforming 3D T1 and the 3D flare. And I think we don't want to be too rigid on this, but you know, I think that's something that we can discuss, and we want to be flexible as we can. But
This is Nina. I would just add for the supplement, if you reread what Cerise sent, it says that prioritization will be given to those that are doing all. So if you aren't doing all, you'll need to talk to us about why that is, and then we'll make a decision on an individual basis.
Yeah, I guess a lot of this could have been lost in all this back and forth, but I really want to make it clear that just because we say something is okay, it doesn't mean it's going to be approved. It's up to the NIA. For this supplement, it's up to the NIA to approve your supplement. It's not up to us. We just certify that you're agreeing to our standards, right? The NIA are the ones you have to convince that what you're doing is scientifically justified. And in the future, you know, I think that could be peer review, right? So it's going to be, it's not up to us to decide these things. Yeah. It's okay with us, but it's not our decision.
Yeah, so right. Let me just, I think I'll just echo what Bill said. So a lot of the questions that have come up about MR have had to do with, you know, exceptions to the notion of single scanner or couple scanners per site, or variations on the MR protocol. And it's not going to be Bill and I who, make the call. It's going to be the imaging committee interfacing with the ADCPIs and with the NIA, who are, going to, I think, determine the answers to these questions, not Bill and I. We're just servants.
Judy Heidelbrink had a question. I need you to unmute your phone or computer.
I think mine was already answered.
Okay, so no hand? All right, cool. Well, I think that's like all the questions and all the discussion time we have. Did you have any parting words, Bill and Cliff?
Well, I just want to reiterate that one of the things we're going to do is we're going to send around a document that we haven't finalized it, but it's going to indicate the things we would like you to agree to for the purposes of the supplement. And you can, check off where you agree and what you're going to do. And that will take the place of this letter of support, just so you know, just to reemphasize how this is going to go. And, I think you all know now everything you need to know as far as what we're going to be looking for. So I don't think there are going to be any surprises.
I just want to comment and thank you both for the thoughtful approach to this and your patience with all the differences among the ADRCs. And I think we've got a few things that we can talk about and try to see what we can do to make this work for the most centers. This has been a great discussion.
Yeah, then I just want to add that, so we definitely got a lot of comments from the different sites immediately after the survey was sent out. And since there's a lot more information that's been provided now, feel free to, send us more emails or use Slack, but emailing is fine as well.
Beth!
Hopefully, yeah, ideally Slack, but if not Slack, email's okay.
Yeah, so thank you to Sterling and Beth and Bill and Cliff. Great job. And Cerise, you're gonna close it?
Well, I think there's lots of follow-up and a lot of details that we're going to work through with the imaging committee. So be on the lookout for more surveys, more discussion about your needs as we all, welcome SCAN into the fold as they're like 2 weeks old. And so it'll be an exciting year to come. So thanks for attending and staying this late. I think we're going to wrap it up. I unmuted Julie Snyder if you wanted to say any closing words, but.
Thank you. I'm having some computer difficulties here. I can't see the Zoom anymore, but I can still hear you. Yeah, so thank you so much, Cerise, Nina, everybody from the NIA. You've made this first virtual ADC meeting quite pleasant, and it really worked out very well in spite of our very rocky preliminary testing day, which we won't tell anyone about. But everyone did a great job. It was really, really interesting to see how everyone's coming together during this time with COVID to really work together, not only on Alzheimer's disease, but also to work together on integrating now the health crisis with COVID. Since it's also affecting mostly our older population, I think there's a lot of work to be done there too. So with that, just thank you everybody for participating and your great talks. And hopefully we'll see each other in October, but more likely it'll be another virtual conference. So, but we'll see. So thank you again, everybody. Thank you, Cerise. Thank you, Nina.
Thank you, Julie. Best wishes, everyone. Stay safe, and thank you.
And everyone who's asking, yes, the meeting is recorded so that we can share it, and we're going to update everything to the NAC website. I don't have any timing because some things need to be edited, so the video editing might delay that. You'll all get a mess a mass message, probably through Slack. So you should probably join it and get all the information. So thanks for joining.
Great. Thank you, everybody. Take
Bye.
care, everybody.